Good morning. Perusing the internet looking for potential topics, I ran across a TED Talk by some fellow named Iliff. And I thought, gee, that was brilliant. I'd love to, wonder if we could get somebody to talk about that at the UW, from the UW. And it turned out he was from the UW. And he was right here across town all the time. Jeffrey Iliff is a professor at the UW Department of Psychiatry and Behavioral Sciences, my own department. He's also a researcher and an associate director of research for the Mental Illness and Research Education and Clinical Center at the VA hospital. And he's also associated with the Alzheimer's Disease Research Center at the UW. In fact, he is the Arthur J. and Marcella McCaffrey Professor in Alzheimer's Research. So it is a delight to welcome him to Horizon House for this Let's Talk Science.
Great. Thanks, Jack, for that kind introduction. So today I'm going to be talking about getting old. You may think that I am not an expert on this, but I find that I am more of an expert every year. And my kids think I am the expert on getting old, in fact. Really, we're going to be talking about a biology that has arisen and has been described over the last 10 or 12 years that is changing how we understand sleep and what its relationship is with dementing disorders like Alzheimer's disease or like Parkinson's. Some things you need to know about me. So my lab's at the VA, so I'm a VA researcher. My lab is funded by the National Institutes of Health. It's funded by the Department of Defense, by the VA, some money from the UW. That's useful to know. I serve as the chair for the scientific advisory board for a company called Applied Cognition. And I do have a stake in that company, so there is conflict of interest that you need to be aware of. I'm a full-time federal employee, so I don't know if that's good or bad in this crowd. But the views today are my views and not that of the government, and I don't know whether that makes that more or less credible. So you can tell me how you feel about that. So we're going to be talking about a general biology, so something that is happening in the brain all the time, something that's happening in the brain when it goes to sleep especially, and we think that this is a biology that's relevant to just normal functioning, to the normal relationship between you and how you think on a daily basis. We think that its dysfunction may have a relationship with a wide range of psychiatric and neurological diseases, from headaches to depression, to PTSD, to Alzheimer's, to whatever. But what I'm going to be talking about today is viewing that disease through the lens of neurodegenerative conditions generally and Alzheimer's disease specifically, right? So I'm going to take a very Alzheimer's-centric focus. In particular, what I'm interested in is the things that are held in common between different neurodegenerative conditions. Right? Everyone has heard of Alzheimer's disease. Everyone has heard of Parkinson's. You may have heard of Lewy body dementia or frontotemporal dementia, or maybe chronic traumatic encephalopathy, which retired football players seem to get. The thing that all of these neurodegenerative conditions have in common is that when you look in the brains of people with them, they have clumps of protein that shouldn't be there. And it's thought that those clumps of protein are what's driving the disease. But it's different clumps of different proteins depending on the disease. So for example, in Alzheimer's disease, there's two things that we mainly focus on. One is clumps of something called amyloid beta in the spaces between the brain cells, or clumps of something called tau that is in the neurons themselves. That's thought to drive the death of neurons and eventually the cognitive impairment that you see in Alzheimer's disease. In conditions like Parkinson's or Lewy body dementia or multiple system atrophy, a different protein, in this case alpha-synuclein, clumps up in the cells of the brain and is thought to drive their eventual death. In ALS, Lou Gehrig's disease, there's still other proteins that seem to clump up. So we're interested in why it is that in some brains and in some people, this process of protein misaggregation begins to happen. Why is that? Why does that happen? one of the clues that we think will help us understand why it happens and what we can do about it is by looking at the features and looking at the experiences that confer risk for these different diseases. So all of these diseases have genetic risks, right? So you might have a familial risk for Alzheimer's disease or Parkinson's disease. And some of those familial risks are very hard. Some of them are, if you have this mutation, you will get the disease. And some of them are, smaller risks. Like if I have this particular gene, I might double my chances for getting the disease. But I'm interested in non-genetic risk factors. Things that happen to you or exposures that you have that change your risk for a disease. And those include things like aging. Aging is the strongest risk factor for all of these neurodegenerative diseases. So much so that you don't even think of it as a risk factor, right? You don't think of, well, I'm getting older, so I have a higher risk. No, these are conditions of the aging brain, right? But it is the aging brain that is the environment where this is happening, right? Other things, cerebrovascular disease, so problems with the blood vessels in your brain that could be connected with strokes, right? Or TIAs, which are transient strokes that people have. Or just general dysfunction of your brain's blood vessels. Having had traumatic brain injuries, whether those are big traumatic brain injuries that land you in the ICU for days or weeks at a time, or whether they're just concussions, whether you played football, whether you were in the military, or whether you slipped and fell on the ice outside, right? Those having had a concussion or having had multiple concussions seems to confer a risk for these conditions. Sleep disruption has arisen in the last couple of years as a key risk factor for these conditions, whether that's untreated obstructive sleep apnea, whether that's insomnia, or whether that's just crummy sleep when you're in your 50s. All of those seem to have confer risk for these conditions. So our question is, what is it that changes in the aging brain? What changes in the injured brain, or what changes in the sleepless brain that makes it vulnerable to this clumping up of proteins, this misaggregation of proteins? And that's really the answer that our lab is trying to understand. And that's where our work over the last 10 or 12 years has taken us. So next, we're going to talk about cranial fluid dynamics with a very smooth transition. When you were having breakfast this morning, you almost certainly did not imagine you were going to hear about cranial fluid dynamics. If you know anything about them, if you know anything about it, this is probably what you know. Your brain is surrounded by fluid. Most people know this, right? Your brain actually sits in a pool of fluid called cerebrospinal fluid. Cerebrospinal fluid it's a relatively clean, clear, protein-free and cell-free fluid. It's produced in the lateral third and fourth ventricles. The ventricles are it's like this system of caves that runs through your brain. The CSF is secreted in that cavernous system. It flows out through those caves, and then it fills the space that surrounds the brain. From that space, it then gets taken up into the blood. And the idea is that the secretion of CSF, it's making in the ventricles, it flowing out through the ventricular system and filling this space around the brain and then eventually being dumped into the blood, that is a one-way path. At least that's what the textbook says. That's if you learned about this in, med school or grad school, or if you in college, if you took a physiology class, this is how we've understood fluid dynamics in the brain for the last 100 years. Things that CSF is doing. Well, one of the things it's doing is it's floating your brain. It's supporting your brain. Your brain has the consistency of slightly cooler than room temperature butter, right? And so if you took a brain and set it on the counter, it would kind of slouch down. And that's not very good for the brain. So the brain actually floats in the fluid that helps to support it. That fluid also helps to cushion the brain from forces. So when you hit your head and your brain rattles around inside your skull, the fluid in there actually serves as a type of cushion. It also serves as a sink for metabolic waste. And when I say sink, I don't mean washing dishes. What I mean is it's a place where things go, right? So there's a model or a hypothesis called the CSF sink hypothesis. That was articulated in the middle of the 20th century by a guy named Hugh Davison. He was in London at the time, that addressed the question of brain waste clearance and how it happens. This model, the CSF sink hypothesis, is based on an anatomical observation. So it's built on this idea that the brain doesn't have lymphatic vessels. So everyone has probably heard of lymphatic vessels. In the rest of your body, you've got your blood vessels, right? They supply oxygen and nutrients to all of the cells of your body and feed them. But then there's this parallel network of vessels that run throughout the body that do two main things. One thing that they do is they help your immune system listen to what's happening in the tissues of your body, right? So it's part of your body's immune surveillance system. The other thing that the lymphatic vasculature does in the rest of your body is it helps to clear away the proteins and wastes that accumulate in the spaces between your body's cells. So these lymphatic vessels actually collect up protein and collect it up into these vessels and eventually dump it into your bloodstream. When your lymphatic vessels get blocked up, bad things happen. So women who have breast cancer, who have to have lymph nodes removed, sometimes you'll end up with lymph blockages where the tissue will swell up because the fluid and the protein in the tissue can't drain properly because the lymphatic vessels have been damaged or destroyed through the process of that treatment, right? Everyone is aware of elephantitis, right? Which I'm not going to show you a picture of it because it's horrendous, but it causes this grotesque swelling of limbs, and it's caused by a failure of the drainage of the waste from between the cells. The interesting thing is, unlike the rest of the body, the brain and the spinal cord don't have lymphatic vessels. And yet the brain makes a great deal of waste. It's very metabolically active. So it's making waste, but then the question is, how does it get rid of it? And so the CSF sink hypothesis states that the CSF space, so the fluid inside your brain and the fluid outside your brain, serves as a sink for that waste. So for wastes that don't get degraded locally, or the waste that can't be dumped into the bloodstream directly, the way that they get out is they exchange out into the CSF that surrounds the brain through a process that is slow and unorganized, but kind of gets the job done, right? That's the CSF sink hypothesis. And that's really where things stood from the middle of the 1950s or so until about 10 years ago. Over the last 10 years, it's been a pretty exciting for the three people in the world who actually care about cranial fluid dynamics. It's very exciting for us. Our understanding of that process has been completely turned on its head. So two big findings have happened. I'm going to tell you all about one of them, and I'm going to tell you this is all I'm going to tell you about the other one. So in 2015, two different groups, one was in Virginia, one was in Finland, described what to all appearances are, and despite what all the textbooks told us, there are lymphatic vessels serving the brain. They're not in the brain, so that actually turns out to be true. It's still, we haven't overturned that yet. There are lymphatic vessels, and you can see them here. So this is a big vein around the brain, and this red thing, these are lymphatic vessels flanking this big vein. So they're embedded in the membranes that surround the brain. But these lymphatic vessels, they serve the brain. So waste from the brain tissue, waste from the fluid that surrounds the brain get taken up into these lymphatic vessels, and they get drained down the lymphatic drainage of your neck. So it turns out that this thing maybe isn't so true. This is really important. It has really dramatic implications for how we understand diseases like multiple sclerosis, right, or neuromyelitis optica, or even Alzheimer's disease. But that's another 2 or 3 hour talk that you guys don't want to hear. The other finding that we've had that began in around 2012. We published the first paper on this. Centers on the question on whether the movement of fluid from outside the brain into the brain and the waste and the clearance of waste out of the brain, is that a process that is unorganized and slow, or is that a process that is actually fast, anatomically organized, and even physiologically regulated? Is it an on-purpose thing, or is it kind of a willy-nilly thing? So this is a pretty straightforward experiment that we did. This is actual data from the paper in 2012. What we're doing here is we're imaging into the brain of a living mouse using an imaging approach called two-photon microscopy. So it's a very expensive microscope that allows you to image through the skull into the brain of a mouse while it's alive. What I'm showing you here, this is the surface of the brain, and then each of these little frames down here is deeper and deeper into the brain tissue, into the cortical mantle. We labeled the blood vessels in red, so we injected a red dye into the bloodstream of the animal, and then we injected a green dye into the fluid that surrounds the brain. And what this shows, it shows, I think, two important things. So first, if I look at the top here, I see that this CSF tracer, it isn't moving vaguely and uniformly over the surface of the brain, but rather it's tracking along the outsides of blood vessels. So you can see it right here. It's following this artery right here. It forms a little halo, right? And it's in what's called a perivascular space. So peri means around, vascular is a blood vessel. So it's the space around a blood vessel. So what's happening is the fluid movement through the brain or around the brain is actually using the blood vessels as a type of scaffold. So it's not in the vessel, right? It's not in the pipe. It's around the pipe. It's as though you took a water pipe, right, that runs under the ground, and if there's a space around the pipe, and if you use that pipe to do something else, like maybe running electrical conduit or something like that, right? So the other thing that you see is that as these vessels dive into the brain tissue, so I can follow this blood vessel, and I can see how it turns and dives down into and through the brain tissue. And you can see a little bit of this little green half moon around these blood vessels. So there's fluid that follows these blood vessels down into the brain tissue. And this green haze that you see in here, that's fluid that started outside the brain and is actually washing through the spaces between the brain cells. So although we thought that CSF was on a one-way trip from the ventricles out into the space around the brain, out into the blood, that's not true. It's recirculating through the spaces between the brain cells, clearing away proteins. This isn't something that's just happening in a tiny, tiny, tiny spot on the brain. This is happening through the entire brain. I bet you didn't know you were going to see a rat MRI. So we have, in our lab, we have access to an MRI. It's a very small MRI. It's actually, it's a very powerful MRI. It's actually much more powerful than the MRIs you would go into, right? But because it's for rodents, it's actually, the bore of it is very small. You couldn't even fit into it. This is essentially the same experiment that I showed you before, except a couple of things are different. First, this is a rat, not a mouse, so it's a little bit bigger. Second, instead of using colored dyes to follow how the fluid is moving, we're using a contrast agent, just like you would have if you went and got an MRI yourself. So we injected the contrast agent into the CSF that surrounds the brain. And then instead of using fluorescence imaging, we're using MRI, which measures the movement of that contrast agent. And so what you can see here, I've labeled how the contrast agent moves in the first hour in blue, in the second hour in green, and in the third hour in red. So blue, green, red, so that you can kind of get a sense of how things move over time. And what you can see is, the whole brain is just sort of red-hued. So what that means is over the course of 3 hours, the fluid around the brain is actually washing through the entirety of the brain tissue, right? It's making its way through. But if I look at where it comes in first, see this green line right here? You see this green line? It's actually, you can see sort of a darkness that is surrounded by two green lines, right? And I can see one here too in this cut right there. What those are the largest arteries of the brain. And so what's happening is the contrast is moving into the brain along the course of these large arteries, and then it washes through the rest of the brain tissue. So the brain is actually repurposing blood vessels whose main job, we thought, was delivering oxygen and glucose and nutrients, right? So delivery. Well, it's actually repurposing those same structures to pump fluid into the brain along the outsides of them. It's scaffolding on them. It's repurposing them for the purpose of clearance of wastes and solutes out of the brain. It's actually super clever, because when you think about the brain is one of the weird things about the brain is it's encased in the skull, right? So there's no space in there. So if you have a brain tumor, for example, one of the reasons why a brain tumor is so terrible is you don't have any extra space in there. And so if a tumor takes up space, that's space that's something else needed, whether that's blood flow or whether that's the rest of your neurons, right? And so real estate in the skull is incredibly valuable. So it's actually interesting that the brain manages this waste clearance by using blood vessels that are already there without having to put lymphatic vessels down into the tissue. It's kind of a clever way to do it. Of course, if this was only rats, you guys probably shouldn't care about it unless I don't know. There's probably academics in the room, and academics care about all sorts of very strange things that nobody else cares about. And so maybe some people care about mice and rats for their own sake. I'm not one of those people. If this is something that's happening in the human brain, if this is something that's for relevant to human health, that matters to me. If this is a funny phenomenon of the rodent brain, I don't care about it. So it turns out that this is something that's happening in the human brain. So almost exactly the same experiment that I just showed you in a rat, we would never do that in a human, but people in Norway will, it turns out. So in the United States, we give contrast agents into the bloodstream, right? And you use it to do all sorts of things. So you can measure leakiness in your brain. You can use it to image different parts of your body. In Norway, there are people who are willing to put that contrast agent into the fluid that surrounds the brain. And so there's a group in Oslo, at the University of Oslo, that's been doing exactly the same thing that we did in rodents a couple of years ago. They're now doing it in people. And the people seem fine, right? At least that's what they say. But we're not doing that here. But the great thing is that they've been able to show that this process of fluid and solute movement through brain tissue is indeed happening in the human brain. So this is human MRI. They've injected a gadolinium-based contrast agent, so an MRI contrast agent. They inject it into the CSF down intrathecally, right? So down by your lumbar spine, near where you would get like an epidural, right? And then they image where it shows up over the next couple hours. And so see this white here, that's the contrast agent filling the fluid spaces at the base of the brain. You see this how the brain tissue is starting to light up. That's the contrast agent moving through the brain tissue. And when the person comes back the next day, you see how the brain here is kind of dark, but the brain here is kind of light? The entire brain is enhancing with the contrast agent. So over the course of 24 hours, fluid is moving into and through the entirety of the human brain. So this fluid flushing is something that's happening both in the rodent brain and in the human brain, maybe all the time. This is a little bit of a Hail Mary pass. So when I talk to graduate students, if I want them to not take my word for it or to dig deeper, I put up a QR code. And I say, pull out your phones, take a picture, and it'll take you to a website that lists all the papers that prove the thing that I'm telling you, because you should not take my word for it. I don't know if this is an audience that does QR codes. Maybe it does, maybe it doesn't. But we can definitely, if this is something you're interested in, I can provide the links to this. But over the last 10 years, we've learned that this process, which we've called glymphatic exchange, right? So gl, because we think it's involves glial cells in the brain, which are the brain's support cells. Lymphatic, because we think it's serving a lymphatic role, so it's glymphatic exchange. So some things that we've learned about it over the last 10 or 12 years. It's anatomically organized and it's rapid, right? I showed you the images, or the movies. It seems to be driven by the pulsation of the arteries themselves. So the reason why it's orienting around the arteries is the arteries are the things that are pulsing. Veins don't pulse the same way that arteries do. We see that it's involved, that the support cells of the brain called astrocytes, their movement of water seems to play an important role in it, and I'll talk more about that in a little bit. We see that it seems to be a feature of the sleeping rather than the waking brain, that this may be actually something that's happening when you sleep. And I'll talk more about that. And actually, so I mentioned before those lymphatic vessels. We think that this exchange and the drainage down those lymphatic vessels, we think that those are actually connected to each other, that it isn't an accident that these two things are happening together. We think that they're actually sort of part of a relay race where this exchange gets stuff from the brain out into the fluid that surrounds it, right? And then the lymphatic vessels pick it up from there and dump it out into the blood. And so it's a two-part relay that gets things out of your brain. You can go to this QR code if you want to see the articles for that, or not. All right. What does this have to do with sleep? What does this have to do with Alzheimer's disease? Well, let's talk about what we know about sleep and Alzheimer's disease first. Okay. So most of us are probably aware that as you age, your sleep tends to get worse and worse, right? You sleep shorter and you sleep just more terribly. right? I find that that's true. I'm about to turn 45, and that's already true of me. I imagine that you could attest to this fact as well, right? It's long also been known that in dementing disorders like Alzheimer's disease, there's a profound disruption of sleep that happens. In fact, it's the disruption of the normal sleep-wake cycle that's one of the things that frequently drives people suffering from these conditions out of a home care environment into an institutionalized home care environment. My mom has dementia right now, and my dad, who is 83, he is her caregiver. And the quality of their life together, right, how they're doing, how she's doing and how he's doing, completely depends on whether she's sleeping well or whether she's up all night, because that has huge impacts on her and devastating impacts on him, right, and on everyone connected to it. So we know that sleep is connected to these dementing disorders. The assumption has always been that, well, the brain is degenerating, right? It's parts of it are dying off or being lost, and it's just that the parts of the brain that control sleep, they're dying, right? And so that's why sleep gets all jumbled up in these people, is because the parts of the brain that control sleep they're not working as well anymore. But there's more recent data that suggests that it isn't just that. It actually might be that poor sleep, especially sleep in midlife, sleep when you're in your 40s and when you're in your 50s and your early 60s, is dictating the very processes that underlie these conditions, and actually might be pushing risk for these conditions. So let's talk about Alzheimer's disease. We mentioned before, Alzheimer's disease is characterized, if I look into the brain of people with Alzheimer's disease, I find two important things. I find amyloid plaques, right? So these are plaques of a protein outside of cells, and I see aggregates of proteins inside cells, right? So a group at the other Washington University, this is Washington University in St. Louis, which is hilariously confusing for people at the University of Washington. So the folks at WashU, over the last 10 years or so, have published a series of studies that show that if you measure amyloid levels, so that's the thing that clumps up in your brain in Alzheimer's disease, if you measure levels in the brain of a mouse, or in the CSF of a human, right? I'm not going to measure it in the brain of a human. That would be unethical. What you see is through the course of a day and a night, amyloid levels go up during waking, and they go down during sleep. They're not constant, but rather in a single day, the levels of amyloid in my brain go up while I'm awake, and when I'm asleep, they go down. And other data that they've shown is that if I stay up all night and measure it tomorrow morning, the drop that was supposed to happen when I sleep doesn't happen, right? So there's this very tight linkage between sleep and slow wave sleep in particular, which is the deepest type of sleep, and this decline of amyloid that's supposed to happen every single day, right? And so the question is, if you sleep poorly or don't sleep enough, and you don't get that decline, is there a consequence of that? Does that promote this process of the buildup of these proteins in your brain? Well, maybe. So there have been a series of studies. I'm showing you one. This is a study that came out of Johns Hopkins a couple of years ago, where they took people, and these are people who are cognitively intact. They're older people, so these are people over the age of 65, but these are not people with dementia. These are not people with cognitive impairment. These are people with fully intact cognition, right? And they asked them, how much do you sleep on average every night? Do you sleep more than 7 hours, 6 or 7 hours, or less than 6 hours? And then they measured how much amyloid was in their brains, right? And they observed that in the people who slept less, so red, hot colors are more, cool colors are less. You can even just see it here. People who report low amounts of sleep have more amyloid in their brain, even before they have cognitive symptoms. And the same is also true, not just of of less sleep, but also poor sleep. So low sleep quality, like I slept 8 hours, but it was terrible, right? And this is one paper, another QR code, and this is something that I'm glad to provide this list. This isn't just one study that has shown this. Now, several studies have now shown, using a bunch of different measures, that people who have poor sleep or short sleep, even earlier on in life, have higher levels of Alzheimer's associated pathology, even before the onset of clinical symptoms. So that has led us to think that poor sleep or short sleep is actually a risk factor for the development of this. And because sleep is a behavior, and it is a behavior that we have some control over, right? If you have OSA, obstructive sleep apnea, you can wear the mask or you cannot wear the mask, right? I'm going to stay up all night and watch movies, or I'm going to go to bed. I'm going to sleep 4 hours a night, or I'm going to work really hard to get 7 hours of sleep a night, right? I can treat the insomnia that I have or not. Like, these are choices and these are behaviors that are potentially modifiable that seem to be conferring risk for the processes underlying Alzheimer's disease and probably other dementing disorders. So what about this brain clearance stuff? I mentioned before that when you sleep, levels of amyloid decline in your brain, right? Why is that? Is it because you're making less, or is it because it's being cleared out more? This is where science is great, because the group at WashU, they think you're making less. That's their thing. The group at UWash thinks it's being cleared more, and the reality is we're probably both wrong, right? It's probably both. But the interesting thing is, when we measure this brain clearance in animals that are awake or asleep, we see that fundamentally there's a fundamental difference in what's happening. So this is the same kind of experiment I showed you before. So this is a mouse. We're imaging through the skull. The difference here is this is a single view down inside the brain. Blood vessels are labeled in blue in this case. And what we did is we injected a tracer into the fluid that surrounds the brain of a mouse while it was awake. So this mouse on the top is awake. It looks like maybe my movie isn't working. It is. It's running. It's just nothing's happening. And then we take the same animal, right? Notice, like, this is literally the same animal 45 minutes apart. And then in this case, we anesthetize it with a cocktail of drugs that mimics slow wave sleep, so deep sleep. We see that the situation completely changes. The same mouse, in one case, it's sedated. In one case, it's awake. In one case, there's rapid washing. And in another case, there's almost none. If we quantify it, this is the difference. This is one of those, like, there's statistics. Statistics, there's lies, there's damn lies, and then there's statistics. This is one of those things where, I can show you this data, but if you just look at this set of movies, you see the difference. And it's not just in this anesthetized state. We also see it when animals are allowed to sleep normally. Under normal conditions of sleep, there's much more rapid washing through brain tissue, and in the waking state, it's much slower. Now, this isn't just the movement of stuff in, this is also the clearance of stuff out. So we can do a different experiment. We can radio label, so we can take radioactive molecules or atoms and put them onto molecules and then inject them into a mouse. And then we can detect that radiation really sensitively to measure how fast stuff gets cleared out. So it's our way of measuring how fast is stuff being flushed. And what we observe is that in the anesthetized or the sleeping animal, so look at this curve here, red and green right here, things get cleared out pretty fast. But in the wake state, they get cleared out more slowly. And we can do some math here and get a measure of how fast it is, and we see that things are being cleared maybe twice as fast in the sleeping compared to the waking state. And that's true of a molecule that we're using just as a regular tracer, and it's also true of amyloid beta. So the brain is actually clearing and flushing amyloid faster during sleep. Now again, if this is only something that's happening in mice, who cares? Is this also happening in humans? Well, let's go back to Norway. Thankfully, the Norwegians, they do crazy things. It's a super clever experiment. It's very simple, but they are basically doing the same thing that we did in rodents, but they're doing this in people. So what they did is they took a group of people, and these aren't just people off the street. These are people who are being evaluated for neurosurgical procedures, right? These are people for whom an injection of a contrast agent into the brain actually is going to give some information that's going to help the neurosurgeon figure out what needs to be done, okay? So they're not endangering people's lives for no reason. There's thought behind this. They're not like unethical European people. But what they're doing is they inject gadolinium into brain tissue in the afternoon, and they bring the people back in the early evening. And they took the group and they divided it into two parts, right? They said group A and group B. And what they observe is if when they image the tracer in group A and group B in the evening, they see about equal amounts of the stuff in the brain. So they got the same amount in both cases. And then group A got to go to sleep at the hospital and do whatever kind of sleep they were able to do, right? So you're being at a hospital. But group B, which is in red, they were kept up all night. And then they imaged how much of the contrast agent was cleared out through the course of the night. And they observed that the people who were kept up all night had a bunch of residual gadolinium, so they didn't clear everything out as quickly. And even when both groups were allowed to sleep on the second night, when they brought the people back after the second night, there was still a difference. So they hadn't recovered yet. There was still a noticeable difference between a group that was kept up for one night and a group that wasn't kept up for one night. So one of the things that the brain is doing when it's sleeping is it's shifting into a kind of cleaning mode, clearing away different wastes, including amyloid beta, and probably including tau and alpha-synuclein, which are the proteins that aggregate in other conditions. Permitting the clearance of things that accumulate through the course of the waking day during the night. So one question is, so if this is something that's happening in sleep, is an impairment of this process part of why the aging brain, part of why the injured brain, and part of why the sleepless brain is vulnerable to diseases like Alzheimer's disease, right? What do we know? Well, we know a couple of things. We know some things from animals, and we're learning some things for humans. So at the UW and at the VA, we have a very, very active program where we're doing actually a lot of this type of work. We're not doing this. We're doing other things where we're measuring this brain clearance in people as they're aging. We're evaluating veterans who've been exposed to blast traumatic brain injury, so men and women both who served in Iraq and Afghanistan who were exposed to IEDs. We see that this process is impaired. We're evaluating sleep apnea, right, and how this affects it. We're evaluating whether Tai Chi actually improves this process. We're doing actually a lot of interesting studies with this. But we've learned a lot from some animal studies over the last 6 or 8 years that has told us about these processes. Answer to the first question is, how about the aging brain? How's this go? So this is a very easy experiment to do. You measure this clearance in young mice and old mice. So a mouse, when it's 2 to 3 months old, so this is a young adult mouse. So this is a mouse with high insurance premiums that's probably making bad relationship decisions. It might still be living in its parents' basement. This 18-month-old mouse, this is late middle age. So this is like, kind of looking at Social Security coming down the road, right? What you can see is in a healthy young mouse, there's really rapid movement of tracer through brain tissue. In old mice, it's dramatically reduced. If we measure the clearance of amyloid, which is the clumpy protein, out of brains of young mice, middle-aged mice, or old mice, we see that in young mice, 70% of the amyloid gets cleared in the first hour. And in the middle-aged mice and in the old mice, there's a stepwise slowing of that process. So in the aging brain, this process of brain clearance is impaired. There's nothing wrong with these mice. These mice don't have a disease. They're just old. There's nothing else wrong with them, right? Other than the fact that they're old. So in stuff that I'm not going to tell you, but I'm glad to share these links with you, we observe that if you give a mouse a traumatic brain injury, right, whether a concussion or whether a big one, this process is impaired. If the mouse has cerebrovascular disease, right, so if it has low blood flow or if it has high blood pressure or other problems with its blood vessels in its brain, if it has strokes, it impairs this process. We see in animal models of Alzheimer's disease, in animal models of diabetes, in animal models of hypertension, even in animal models of headaches, migraines, we see that this process is impaired. And early studies are now showing that this process seems to be impaired in early human studies where we're starting to evaluate this. So groups, including ours and including some others kind of around the country and around the world, are starting to measure this in people now. It's new, so we've only been doing this about 10 years, and we've only been imaging humans for about the last 4. But we're starting to see evidence that this is part of what's going on. So what's changing? And here we're going to get a little nerdy, all right? So we're going to nerd out a little bit. I'm going to tell you about some molecular cell biology that I think is important. And I think it'll make an intuitive sense. So what's changing the aging brain or in the injured brain that's underlying this impairment, okay? And a thing that's changing is this thing, the purple thing. This is an image from a slice of a rat brain where we've labeled the support cells of the brain in green. So these are the star-shaped cells called astrocytes, right? You can kind of see why they're called astrocytes. And we labeled this protein called aquaporin 4 in purple. Aquaporin 4 is a protein that lets water move across cellular membranes, right? And if I look at where the aquaporin 4 is in this brain, you might look at this and say, well, it's in the blood vessels. Can't you see that? It's obviously in the blood vessels, but it's not. It's actually in the astrocytes, but it's in the part of the astrocyte that faces the blood vessels. So if I'm a blood vessel looking out at the brain, right? You have hundreds and hundreds and hundreds of miles of blood vessels in your brain. So every one of those, when I look out, I don't see neurons. I see astrocyte processes that surround more than 95% of the surface area of the blood vessels. Right? So the blood vessels are surrounded by astrocytes, right? And as I look at that 95%, as I look at that astrocyte, 50% of the surface area that I see is this water channel. So that means that that part of the astrocyte is specialized to move a whole bunch of water. It's specialized for water movement, which is weird because your brain blood vessels, guess what it's specialized to do? Not move water, right? In your blood vessels in the rest of your body, in your legs and in your heart, blood vessels move water very easily. They're very leaky. In the brain, they're very tight because your brain doesn't like having extra water in it, right? That's edema and that's high pressure, right? So it's as though I had a house and I built a door that opens up into a brick wall, right? Why would I do that? Why would evolution or like whatever, why would that process arise to have one surface that's specialized for water movement abut up against a surface that is made not to move water? Unless the brick wall that I look at is a building and there's an alleyway that goes right this way, right? So that's what we think aquaporin is doing. We think what it's doing, it's not moving water in and out of the blood vessels. We think it's letting it into those peривascular spaces and that it's organizing water movement through the brain tissue along the scaffold of the blood vessels. So it's organizing water movement through the brain along blood vessels. Well, we can test that. We have animals. The nice thing about mice is it's actually relatively easy to change their genes. And we can make a mouse, or other people can make mice, where they don't even have this aquaporin 4. So we can make a mouse that doesn't have it. And what we observe is that in that mouse, in a normal mouse, you get tracer movement, CSF movement into the brain tissue. In the mouse without aquaporin 4, very little. If we measure the clearance of stuff out of brain tissue in a normal mouse, a wild-type mouse with normal genes, we see that things get cleared with a certain rate, like amyloid gets cleared with a certain rate. If we delete aquaporin 4, if we get rid of that gene, everything's slower. So this water channel, we think, organizes fluid movement through brain tissue. If you get rid of it, you get slower amyloid clearance. So does that mean that when people get old or when you have a brain injury, do you lose your aquaporin 4? Is that what happens? Well, it turns out that was our hypothesis, but that's why you do the experiment. It turns out, no, absolutely not. Actually, you get more aquaporin 4, which was very confusing. But what happens is the aquaporin loses that very fine localization. So remember in that image, almost all the aquaporin 4 was along the blood vessels, almost all of it. So if I look in a healthy young animal, like this is a blood vessel, almost all the aquaporin 4 is right here in this astrocyte. In the older animal, I get tons of aquaporin 4, but it's everywhere. So what we think is happening here is this cell, because it now has this water channel everywhere, it's as though your house that had one door now has 1 million doors. And the doors open up everywhere. And so now you're not just having people go into that alleyway, you have people going into your backyard and into your basement, and they're going all over the place. And so now the system doesn't work as well. Well, we can do an experiment that can test that hypothesis. Is that true? Is where this water channel is it that important, or is it not important? So we know that aquaporin 4 is by the blood vessels because it anchors to a bunch of other proteins. There's a protein that holds onto the blood vessel, and then there's another protein that holds onto it, and another protein that holds onto it, and another protein that holds onto it, and then that holds onto aquaporin 4. So there's a chain of proteins that hold aquaporin 4 where it's supposed to be. And if we get rid of any of these, and I'm going to focus in on this guy right here, if I get rid of this one right here, I get aquaporin 4 at normal levels, so I have normal amounts of it, but it's everywhere. Well, in that animal, I can measure this exchange by MRI. So if I inject contrast agent over time, you see enhancement of the brain as the contrast agent comes through the brain tissue. If I get rid of the aquaporin 4 from the blood vessels, it gets slower. So I slow this washing if I just change where that water channel is. I can do something else. I can take that animal and so we have mice that spontaneously make amyloid plaques. So these are mice that make the same pathology that humans get in Alzheimer's disease, right? They're genetically manipulated, so they develop it over the first couple months of their life, so they get it real early. But I can take that mouse and I can breed it with the mouse that doesn't have my gene of interest, and then I can measure how many plaques does it have. And you can look here, in a normal mouse, you get some plaques. But if I get rid of the normal placement of that water channel, I get tons and tons and tons of plaques. So many plaques. So in aging, we lose this water channel's proper place. And when you lose the proper place, it impairs cleaning and promotes amyloid. Okay? These are mice. Who cares about mice? Do you guys care about? I don't care about mice. You shouldn't care about mice unless it's relevant to humans. What do we see in humans? So this is a case series that we did in collaboration with the Oregon Brain Bank down at the Portland Alzheimer's Center, where we looked into the frontal cortex of people who donated their brains at their death for this kind of research so we can understand these processes, right? So we looked into the cortex of people who were cognitively normal. So these are people who died without cognitive impairment and without Alzheimer's disease. And we looked in people who had Alzheimer's disease and people who had mild cognitive impairment, which is kind of an in-between zone, right? And we saw that indeed, people with Alzheimer's disease had less localization of aquaporin 4 where it was supposed to be. So their aquaporin 4 wasn't in the right place. So people with Alzheimer's disease have these changes. We also observed that if we measure cognitive decline in people before they have Alzheimer's disease. So these are people who are not demented yet. These are people who are cognitively normal or have MCI. And we measure where the localization is and how their cognitive function is. We see that the worse the localization, the worse the cognitive function. And on this scale, going up is bad, and on this scale, going down is bad. So even in people before they're demented, changes in localization are associated with changes in cognitive function. When we measure Alzheimer's pathology in those same people, we see that changes in localization are associated with Alzheimer's pathology in people. So we see evidence of the same changes that definitely cause things in mice. We see the same associations in humans. Okay? Okay. That was a lot of nerding out. We're going to review, then I'm going to talk about why this matters. And we're going to have questions. First, brain clearance, the glymphatic system. So during sleep, the brain is washing fluid through brain tissue, supporting the clearance of all sorts of things, including the things that clump up in the setting of Alzheimer's disease or Parkinson's. In animal models, we see impairment of this process associated with all the risk factors for Alzheimer's disease. And we see, if we impair this experimentally, we can drive the production of pathology. Does that prove that this is actually underlying Alzheimer's disease? No. We don't have that smoking gun. But to the extent that we can evaluate this in living people, right, we do see corroborating evidence. So we're really starting to think that an impairment of this process may be one of the things that links things like aging, cerebrovascular disease, traumatic brain injury, and poor sleep to diseases like Alzheimer's or vascular dementia or Parkinson's. Okay? Who cares? Well, who cares? Everyone here is familiar with Alzheimer's disease. Everyone knows people who have or had dementia, have it, or certainly are going to have it, right? And that's true of all of us these days. The trouble is, and everyone's familiar with the new Alzheimer's treatments. So lecanemab, it's an immunotherapy that targets amyloid plaques and pulls them out of your brain. It's really good at that. It's really good at getting amyloid plaques out of your brain. It's not as good at preventing cognitive impairment, right? It may help, may not. It's unclear if the risk is worth the benefit in everyone, right? But it's FDA approved now. So that's an approach called secondary or tertiary prevention. So it's the pathology's there, we're going to get rid of it in hopes of stopping the impairment, okay? But we know that amyloid and that tau, it shows up in the brain beginning 15 or 20 years before the onset of clinical symptoms. So when you end up at the doctor's office with cognitive symptoms because you're not remembering things properly or losing your way, by the time you get there, that pathology has been at work for 20 years, 15 years, 10 years, right? That's what the data is telling us, that it's sort of in people just silently at this point, and those are people that will eventually develop cognitive impairment. And so the goal with these antibodies is, hey, if we could identify people who aren't demented yet, don't have cognitive impairment yet, but they have the amyloid, if we get the amyloid out, could we prevent dementia? Could we prevent cognitive impairment? That's where the Alzheimer's field is at this moment, right now, is how do I identify people at risk and prevent it? But, if an impairment of this process is what's causing amyloid deposition, if this is what's causing tau, then if you could identify people who have impaired function, you could identify people at risk of getting amyloid before they get it, right? So think about heart disease. If you have a heart attack, the good news is you go to the cath lab and treatment for a heart attack is better now than it was 20 years ago, right? Better now than it was 40 years ago, thank goodness. But heart disease hasn't gone down the killer list because of cath labs. It's gone because everyone has aggressive treatment of hypertension and hyperlipidemia. So we know that hypertension leads to blood vessel disease, leads to heart disease, leads to heart attacks. We know that. So that if we identify hypertension, we can treat that and we prevent the heart disease, prevent the heart attack. Primary prevention. You detect the thing that gives you risk to prevent the pathology that causes the problem. In Alzheimer's disease, if we can identify the thing that puts you at risk of the pathology of Alzheimer's disease, we can maybe prevent that pathology in the first place so that people at risk would never get the amyloid plaques, or if they got them, they'd get them later, right? So you don't have to treat the amyloid and you don't have to treat the cognitive impairment because you prevent the whole thing in the first place. It's the heart disease model but applied to Alzheimer's disease. So if we had, say, MRI approaches or a device or a biomarker that allowed us to know, is your glymphatic system impaired in your 30s or 40s or 50s, then we could say, great, well, we have a drug that we can give you, or we have a device we can put you on, or we have something we have an exercise we'll ask you to do that will keep this process going, preventing the development of the pathology that is Alzheimer's disease and preventing the eventual cognitive impairment. That's our model. That's why it matters. Okay? So, new science, new biology, new reason why we sleep, and now we're starting to understand a role that it might play in the development of human Alzheimer's disease, and we're starting to think about what we can do with it. What can we do to actually change the course of the disease, not by pulling amyloid plaques out of your brain and hoping things go well, but actually by preventing it in the same way that we do for things like heart disease. So, glad to take questions, and thanks. Yes.
What does taking a sleep aid do to the quality of sleep?
So it depends on the sleep aid. This is actually a really difficult thing. So many people take, for example, Benadryl or we'll take like a cold medicine, like an antihistamine, to help them sleep. And they do that because it's over the counter, and they do that because, well, I've done that, you're like those are safe in pregnancy. So a lot of people did that when they were not sleeping well when they had babies. Like people have been doing that for a long time, but there's actually now data that have come from a study here in Seattle suggesting that antihistamines in particular may increase your risk for dementia, right? So it matters first what kind of sleep different things give you. So as an example, some people drink alcohol to help them sleep. I'm not saying that that's anyone here. The type of sleep that you get that's induced by alcohol isn't very good sleep. It's poor in quality, and it doesn't have the normal sleep structure. Ambien, which is a commonly used sleep aid, may not produce the right balance of sleep, right? And so the question arises, what is the right type of sleep that we need to get? And we actually don't have a good answer to that. So there's a new set of drugs that are out. They're called orexin inhibitors, or DORAs. They seem to be maybe beneficial in this space, but that data is very early. But the big thing is some sleep aids are associated with actually increased risk for cognitive impairment and dementia. So those are things you should be definitely talking to your PCP about. And then other ones, the jury is still out on whether they generate the right types of sleep to give you healthy sleep.
So much of the research has been done on men. We found that in the heart.
Yes.
And I wonder, are you able to break down men and women in this, or are we all just finding out what men are doing?
Well, and we're
and you are at the VA, and I work there, and there were a lot of men.
there are a lot of men at the VA. That's true, and specifically white men in our Alzheimer's Center cohorts, especially, right? White, highly educated people, right? Not normal. Not that highly educated people aren't normal, but they're not, right? So at this point, there no sex difference in this biology has yet been observed. But no one has asked the question really, really hard. We have a current study that that has not yet been published where we do see sex differences in this. And we see sex difference. So we have a drug that is currently, it's FDA approved, it's being used for something else that seems to enhance this process. But its effect is different in male and female mice, right? We do know that there are differences in risk for Alzheimer's disease between men and women. We suspect that perimenopausal changes in sleep and cognition might have something to do with this biology, right? But no one has studied it because no one wants to study menopausal women. No, that's the problem with science is they don't, it has been a subject that no one has been interested in historically. And as a result, we know nothing about something that when I talk to people in that age range, they're like, oh, that's a thing. And yet, no, there's no study focusing on it. So,
I have two questions. One, what does your company, Applied Cognition, do? And two, could it be that many of the processes that you described are a function of something else, like what's going on in the genes? And if that were the case, you could begin treating, quote, Alzheimer's way before people have any symptoms at all.
absolutely. So the company that I work with, it's called Applied Cognition. It's a startup out of the Bay Area. It has a wearable device that detects glymphatic function in the human brain. So we just finished collection on our first big study in human participants out of a big retirement community in Florida. We finished it this June, so we're about to publish those data. The idea being, can we measure this in people in the natural world as opposed to in an MRI scanner, right? So that's part of what this company does. The question of risk and genetics, so we see clear genetic associations. So there are naturally occurring gene variants in the aquaporin 4 gene, and then all the genes of the proteins that are associated with it. And we see that those variants are associated with risk of Alzheimer's disease. So when we look there's some big genetic risk factor studies that have been done. So we can look into that data and identify risk for Alzheimer's disease that connects with those. And so yes, it may be possible to do a genotype on you to say, wow, your system is predisposed to failure, or it's predisposed to function less well. And so as a result, you should be monitored more aggressively in the same way that right now you can get your ApoE4 genotype, and that tells you a lot about your Alzheimer's disease risk. Maybe 50% of the genetic risk for sporadic Alzheimer's disease connects to ApoE4. And it's possible that there are gene variants in these astrocytic genes that may confer risk also. So,
I wanted to thank you for opening a door that I've been staring at for years. And I want to also clarify that I understand this. I'll just speak. I happen to have had a PET scan. I have been told that I have elevated amyloid. I've also been told that I'm cognitively normal today. I don't know about tomorrow. Yep. But, and also have the ApoE4 risk factor. So here's what I'm hearing you say, that the crucial thing is not the existence of amyloid beta, but the glymphatic impairment. Is that correct or not?
So I would not say that.
Okay,
I would
please
say
explain.
in Alzheimer's disease, one of the crucial things is the amyloid beta. And so, and this gets at therapeutic strategy. Just like if you have a heart attack right now, I'm not going to say, we should really do something about your hypertension instead of taking you to the cath lab, right? You need to go to the cath lab. Once the amyloid is there, the new the new drugs are trying to address the fact that it's there, and they're trying to stop what comes next, which is the cognitive impairment, right? Where we think there's an opportunity here, and this is the unfortunate, when I think about myself, I actually don't, the things that we're currently doing, I actually don't think they're going to help me. And I don't think, they're certainly not going to help my mom. I think that these are going to be things that will change how my kids deal with this in terms of like a prevention strategy, right? So can we identify risk in, when you're 40 and prevent the amyloid from getting there when you're 60 or 70, right? That's the goal. The downside to that is, what about us? Well, we need better drugs. There's new drugs. There's things now, which is good because there didn't used to be anything. Now there's something, and that's better. That's certainly better than nothing. But where the real money is, not the money, but the power, where the leverage is can you prevent it in the first place? Can you prevent the amyloid in the first place in people who are right now 30 years old or 20 years old? And I'm so far past that that this is going to be for them, right? Which is great.
In your earlier slide, you showed a correlation of cognitive decline with the amount of sleep.
Yep.
How do you quantify the sleep? Can the sleep be continuous, or can it be the same as if it were in, stages of?
Yeah. So part of the problem with that is the measurement of sleep is not done well. We're publishing a paper. It comes out on December 4th. So it's like embargoed right now, so no one's supposed to know about it. But what we show is in a group of Seattle people, like part of the Seattle longitudinal study, if we just asked them, hey, in an average month, how much do you sleep a night? And you tell me, 5 hours, 7 hours, 9 hours, whatever. And if I measured that twice, right, over the course of 10 years, right, 2 times, a person's going to be flat, they're going to go up, or they're going to go down. And so I could classify people as increasers, decreasers, or stable, right? But in this study, we actually measured people 5 times, right? So we actually have 5 measurements of sleep in these people, where we're not measuring sleep. All we're doing is asking them about sleep. But one of the things that we see is that the slope actually doesn't make any difference. It's the variability, right? And so what that tells us is, in studies that have told us about when we look at sleep, very frequently we're looking at sleep at a single time point, and very frequently we're only looking at like what you say about your sleep. And frankly, what people say about their sleep becomes less and less reliable the older you get. The data bear that out. And maybe that's because you become liars, or maybe that's because you just don't notice, like you don't notice bad sleep when you have it, right? We don't know why that is. And so the measurement of sleep in the studies that we're talking about is done poorly. Very frequently, these studies It's looking at old data. It's looking at data that was collected 30 years ago. Because if I'm measuring risk of cognitive impairment, I have to measure that over the course of 20 years. And so basically the data I have today are questions that I asked 25 years ago. And 25 years ago, no one thought that sleep was connected to Alzheimer's disease, right? And so no one was asking good questions about sleep, and certainly no one was measuring sleep with devices. And so now what we have are good measurements of sleep, and now we got to wait 30 years for those people to get older. And so we're sort of in a place where we don't have good data.
Question here. If we, at our age, people in their 80s and 90s, suddenly began sleeping better, would that help in terms of slowing the advance of dementia?
Yeah, so that's a great question. The honest answer is we don't have the data for that. But does anyone here think that if you got better sleep, you wouldn't do better, or if you got worse sleep, you wouldn't do worse? Does anyone think that that's true? Right, which is like the ask your grandparent test of something, right? We do know in sleep apnea, which is the one thing we do have good data on. Sleep apnea is a really interesting condition where a sleep problem is caused by something that can be cured with a device. Now, many people don't like the device. Many people don't use the device, right? But when you use the device, it cures the condition, sleep apnea, which results in bad sleep. We do know that that people who use CPAP, it has, after the first night, it changes your amyloid levels. First night. It's a huge thing. It has clear effects on cognition, right? We don't yet have data on whether it changes risk of dementia, but no one thinks that it's not gonna. Right? We just don't have the data yet. So sleep apnea is an extreme case because when you have sleep apnea, your sleep is particularly bad, right? And it's a case where you can literally turn it on and turn it off. But most of us think that good sleep, at the very least, it may not change, so the amyloid that you have and the tau that you have, it's probably not going to change that, right? Because those are clumps that are going to be, they're there. But the functioning that you have in the face of that pathology, your reserve is probably higher. So I see this with my mom. There was a period she had an infection and then she didn't sleep for like a week and her cognition, the floor dropped out of it. She went from being pretty normal to being completely like disconnected and completely like unattached to anything. Working with her PCP, getting her medication, getting her sleep medication, she's on trazodone, getting that right, getting her onto a sleep schedule and a routine, restoring that, her function came back. Did her pathology change? No, almost certainly not. But her brain's function in the face of that pathology, its resilience changed, or its reserve changed. And that's something that, when you're in your 80s, changing your sleep now, that's probably where you're going to see the effects. And do you care whether that's a pathological change or or do you just care, are you better? Right?
We're going to take two more questions, one there and one back.
Yes, you talk about healthy sleep, and I actually went to a sleep study and was told that your sleep is the way it is because you're old. And that was not a very helpful suggestion. And so, but I do use a Fitbit, and it shows deep sleep, light sleep, and REM.
Yep.
What's healthy?
It is true that older people just have worse sleep. So we have studies that we're doing right now where we're intervening to help sleep, and then people say, well, you're helping people who don't have clinical sleep problems, but they do. They're old. Like that's their sleep problem, and old people have bad sleep. Like that's just true. They have shorter sleep, they have more fragmented sleep, and they have less both N3 deep sleep, and they have less REM, right? So good sleep, healthy sleep, is both the right quantity, so that's 7 hours, right? And it's the right type. So during the night, you go through different sleep stages and you cycle through those different sleep stages. At the beginning of the night, your cycles include more deep sleep, And at the end of the night, your cycles tend to include more REM sleep, which we think of as being dream associated, right? And so good sleep includes all of those in their proper proportion, in their proper amounts. The trouble is, when you get old, it's harder to get that. Now, the common wisdom is, well, old people just don't sleep. They don't need it. They seem fine. Maybe. It's probably not that you don't need that much. It's probably not that you need less sleep. It's just probably harder for you to get it. And you just figure out how to cope and just sort of do life without it, right? But for me, I noticed that like me staying fit and not like chubby, it's harder for me at the age of 40 than it was when I was 22. It is just harder. That doesn't mean it's less important. It just means I have to work harder. Now, me sleeping well it's easier now that my kids are moving out of the house, but it's harder as I get older because I have to get up and go to the bathroom more and I just don't sleep as well. Doesn't make it less important. It just means I have to work harder to do it. And I have to like organize my life in a way to, trick it into doing it the right way, right? It's harder, but not less important. Does that make sense?
Last question.
Yes, you mentioned Tai Chi just briefly. I want to skip over that, but it made me think of meditation and wondering if you've done any studies, if it could possibly be a supplement to good sleep.
Yep. So there's clear data. There is good data that suggests that people who are active in exercise sleep better. People who meditate sleep better. People who do Tai Chi sleep better. People who do yoga sleep better. Do they all have the same active ingredient, which is just giving a rip about your body? Maybe, right? Like, because it's also true that the people who meditate frequently eat better. And they do other things, right? So is it the meditation? Maybe. We focused on Tai Chi because it has a mindful element to it. It has a breathing element. We think actually breathing is really important. And it has a movement and also a social element to it. So it's sort of tapping into a lot of different things. What the active ingredient in it is it the mindfulness? Is it the breathing? Is it the exercise? Or is it all of them? That study can't get at that. But so there's clear effects on sleep of those things. Whether it's just healthy people do those things, or whether it's those things actually help health, we don't have a clear answer on. But in general, most of those things we think that they will prove beneficial with this biology. So,
This has got to be the only lecture I've ever attended where if you slept through it, it might be good for you. Wonderful. Jeffrey Iliff, thank you very much. Wonderful.